Steve Scheer, and I'm introducing today's panelists who are going to discuss gene therapy and drug development for CMT type 2. Could I have the second slide, please? So it's my pleasure to introduce Jim Wilson, who is a professor also at the University of Pennsylvania and who wears many hats. He's also the head of the Orphan Disease Center, and he runs a large gene therapy program and has founded a company or two that's devoted to gene therapy, and Passage Bio in particular is interested in CMT2A. Luke Judge is an associate professor of pediatrics at UCSF, that's University of California, San Francisco, and he works at the Gladstone Institute, which it happens to be the place where CRISPR-Cas9 was basically invented. And you'll be hearing from Luke about progress that's being made for using that technology to treat CMT type 2. Last but not least is Robert Burgess, who's the principal investigator at the Jackson Laboratories. And I've known Rob a long time, and he has worked on animal models of CMT and is now leading investigations into therapeutic treatments for CMT, and on top of his elegant investigations as to the pathogenesis of different kinds of CMT. So we're going to hear from each of these speakers in order. Each person will speak for about 15 minutes. If there's time left over, we'll have the chance to take a couple of questions, I presume, but it's not necessarily going to work out that way. So thank you for coming to our session, and I'll turn it over to Dr. Wilson.
Steve, do you see my first slide? Okay, great. All right, it's a pleasure to be here and to meet with the patient and the families to talk about our work in gene therapy. Before I launch into my 15 minutes, I'd like to acknowledge the many sponsors and scientists in core facilities at the University of Pennsylvania that have made this possible. In particular, Christian Hinderer, who's in charge of the CMT project, sorted by Cecilia. We've been interested in developing gene therapy for many diseases, but in particular, the monogenic CNS diseases, which bear a disproportionate burden of disability and mortality. And monogenic CNS diseases have been the focus of many different academic labs as well as companies. Just would like to give the audience a brief primer on gene therapy and how it works. This slide illustrates the basic foundational technology of using a virus as a delivery vehicle for gene therapy. In the upper left, we show a virus with a genome inside that can deliver that genome into a cell to express an RNA and then express a protein. The key to the success of this technology is to render the virus defective so it does not cause any toxicity, but still is able to deliver its payload. The viruses that have been developed for gene therapy primarily are based on a family of viruses called adeno-associated viruses, a picture of which is shown here. And there are a variety of different forms of these viruses, but they've generally been shown to be safe. They're moderately efficient. So we're going to talk about the use of these viruses, AAV, for gene therapy for CNS disease, in particular CMT2A. The first application of one of these so-called vectors was based on technology that came out of my lab around year 2000 in a system called AAV9 that was used to treat on neurodegenerative disease. But to illustrate the power of this technology, I'm going to share with you experiments in a dog model pointing how and where AAV can deliver its genes. One of the challenges in delivering genes to the brain or the spinal cord is the blood-brain barrier that prevents vectors from getting on the other side of the blood vessel. In this particular application, an AV9 vector expressing a normal version of a gene for the disease spinal muscular atrophy type 1 is injected into the blood and hopefully then transported on the other side to target the relevant cells. When this was done at Penn and animal models, as shown here, that the vector was efficient really only in motor neurons of the spinal cord, but not in other parts of the brain. So an intravenous delivery of AV9 would not be good for most neurologic diseases, but possibly for certain diseases that are caused by pathology in the spinal cord. This vector was transferred to an academic lab at Nationwide Children's, and then ultimately to a company, and was developed for the treatment of a disease called spinal muscular atrophy type 1, which is a neurodegenerative disease in which babies born with this disease do not gain any motor functions and die usually between 1 to 2 years of age. The root of the disease in SMA type 1 is similar to certain types of CMT that affect the motor neurons that emanate from the cord and innervate the muscle. A deficiency of the gene SMN1 leads to degeneration of these nerves and therefore loss of motor function. It was shown by the group at Nationwide that an intravenous infusion of this vector in kids with SMA1 before they progress too far have almost miraculous results where the kids survive and they were able to achieve many milestones. This was then developed as the second approved gene therapy for any genetic disease, and now is available throughout the globe and is standard of care for treating this disease. I point that out because it illustrates the potential of gene therapy for an efficient and long-lasting effect. However, the amount of vector that's necessary to target the motor neurons of the spinal cord in that application is very high and difficult to achieve in older patients. So we began to consider alternative ways to transfer genes to cells of the brain and the spinal cord in a process that is similar to a lumbar puncture where the vector is infused into that space where the cerebral spinal fluid is, but closer to the brain rather than down in the spinal cord. We call this an Icm injection. Colleague of mine, Ron Crystal, demonstrated the distribution of an AAV9 vector when injected into the cisterna magna in non-human primates or monkeys, in which he labeled the vector and followed its distribution throughout the brain over time and down the spinal cord. And it definitely was an efficient way to get this distribution of the actual vector. But the question is, does the vector deposit its genes and express those genes in the right cells? And in fact, shown in non-human primates, this approach will correct genes in this part of the spinal cord where the motor neuron cell bodies are, and also the sensory neurons and this shows gene transfer in a peripheral nerve of a non-human primate. This kind of profile is exactly what we would be looking for certain forms of CMT. So in contrary to many diseases where gene transfer may not be sufficiently efficient, it seems to me that several types of CMT may be well positioned to move forward with gene therapy approaches. So the approach that we proceeded with input from Steve, who's a colleague of ours at Penn, as he noted, was to select CMT2A as our first approach and hopefully leverage this experience across many other forms of the CMT. As many of you know, it's caused by mutations in a gene encoding, we'll call it MFN2, protein required for protein fusion. most cases are caused by a single amino acid mutation that leads to a dominant or gain of function a mode of action. As a result of this mutation, there's toxicity to the lower motor neurons and the sensory neurons, again, the areas where our vectors can target. Our gene therapy strategy is to develop a vector capable of interfering or downregulating or neutralizing this gain of function mutation and injecting that into the cisterna magna to target the motor neurons and the primary sensory neurons. So here, in contrast to many other forms of gene therapy, such as SMA, where we have to replace a function, here we have to downregulate a function, which is a little more complicated. And we'll tell you about the approach that we've taken in the next slide. But important to any therapeutic intervention is the evaluation of this approach to determine whether there's demonstration of effectiveness in animal models. And there are a variety of great animal models which we are pursuing, one of which is a mouse model that we've evaluated in collaboration with our colleagues at Jackson Labs. This mouse model was described in 2019, and it's a mouse in which the human version of this gene with the pathogenic mutation is introduced in the germline of the mouse. A control is a normal version of this gene. So this is the control mouse, the normal mouse, and then the animal that is expressing this pathogenic version. You can see from this picture that that expression of this gene in the neurons of this animal leads to an effect in which it is doesn't gain as much weight or grows, basically failure to thrive. This just shows the impact of the mutation on the weight of the animal over time. And these are the control animals, and this is the transgenic, and this is from the paper that was published in JCI. And we're using this as a readout to determine whether gene therapy works. So the other readout is an evaluation of motor strength in something called the grip test, where the animals have a weaker grip, as shown here. So we've developed an approach for gene therapy in which we overexpress a normal version of the MFN2 gene, but also considered a way in which we not only overexpress the normal version, but we incorporate a cassette that can specifically downregulate the endogenous gene without impacting on this normal version. So this is our vector strategy. Again, two approaches to downregulate specifically the pathogenic protein. So we conducted some pilot experiments in the MFN2 mouse, again, in collaboration with our colleagues at Jackson Labs. We first look at the weight and that the untreated but mutant mice do not gain as much weight as the normal animals. And the vector-treated animals, though, we were delighted to see that gain weight almost as much as the normal animals. We also, in our first experiment, evaluated the impact of this on survival. And the way that this experiment was conducted, the mutant animals that are untreated realized some mortality within the first 30 days. But when we introduced vector into those animals, they did not, they were able to survive. So we had evidence based on weight gain and survival that this may be effective. And then we did one other test, which is the grip strength test, which is shown here, where the animal clings on to this this cage and hangs on until it can no longer do so, measuring its motor strength. And this is the untreated animals. This is the mutant animals, where it has a lower grip strength, and the treated animals, which are statistically improved, not quite normal, but much improved. So where are we? Just to summarize, we are now moving into non-human primates with this clinical candidate vector. We've dosed one animal and as a pilot experiment, and so far the animal did well during its in-life phase. So just to summarize about this procedure, which is the ICM AEV injection, which was pioneered by a colleague of ours at Penn, Brian Pukamis, and he has become the world expert in doing this safely. And this just shows an example of a CAT scan that is done while the needle is infused into the cerebral spinal fluid. So running out of time, but I just want to summarize what our experience has been in clinical trials for treating neurologic diseases. There are about 21 gene therapy trials that are in the clinic now, and 7 of which use an ICM infusion. Our experience at Penn is we have extensive experience with the ICM infusion in monkeys. We've dosed this in 485 monkeys, 10 different programs. There was evidence of pathology that was non-clinically significant. And importantly, Penn has now supported 5 active clinical programs using this approach, and we hope CMT-2A to be one of those. So I think I will no longer share my screen, and thanks for the invite to be here.
Thank you, Jim. So I think, Luke, you're next. You're muted.
All right, thank you. All right, hopefully everyone can now see my slides. So thanks for the invitation. It's my pleasure to be able to speak with you today about the gene editing therapy that we're doing for type 2 CMT. And I think we got a perfect introduction from Dr. Wilson into gene therapy in general. I have a quick slide here sort of briefly comparing the differences between the type of gene therapy strategy that Dr. Wilson's group is using, which is shown on the left here, as he so nicely described, which is a strategy where you use a vector typically derived from a virus that packages artificial copies of a functional gene that replaces the function of a mutated gene that is essentially not working. And so these are perfect for diseases in which the patient does not have any functional copy of disease or any functional copy of a gene, which causes the disease. And just to point out that in the case of this strategy, you want the cargo that is delivered by this vector, this artificial gene sequence, to stay in the body and persist as long as possible, ideally for the person's entire life, so that they continue to have the benefit. In contrast, the type of gene therapy that we are developing, which is based on CRISPR gene editing, involves delivering an enzyme called a Cas enzyme, typically, that has also uses a very short artificial genetic sequence to target it to the person's own genetic code in their DNA, where the Cas enzyme acts like a molecular scissors that cuts the DNA into 2 pieces that then goes on to be repaired. And in that process, permanent changes are made to the person's own genetic sequence. So in its simplest form, this strategy is best for what we call dominant diseases, where the patient does have one functional copy of the gene, but they have a mutated gene that actually interferes with the function of the normal gene. And so in its simplest strategy, we use this CRISPR gene editing to actually knock out or disable the mutant copy of the gene, which then would allow the normal gene to function and no longer cause disease. And in contrast to gene replacement therapy, in this case, because we're changing the person's own underlying genetic sequence, we want this CRISPR enzyme to only be existent and active in the body for the briefest amount of time necessary, and then we want it to go away so that it doesn't cause side effects. I also just want to start by acknowledging that it's quite remarkable that CRISPR gene editing was really first described less than 10 years ago here by our colleague in the San Francisco Bay Area, Jennifer Doudna and her collaborators and her group in the laboratory at UC Berkeley, and she also has a laboratory at the Gladstone Institutes in San Francisco, where I work. And she's gone on to found the Innovative Genomics Institute, which is a UC Berkeley and UCSF institution that is mission is to translate CRISPR gene editing into technologies to improve human health. And in this short amount of time, we're already seeing many different clinical trials testing CRISPR gene editing in humans for a variety of diseases. And I've highlighted just a few of those that are very exciting here. One of those being gene editing for sickle cell anemia, which is being done by my colleagues in the Department of Pediatrics at UCSF as well as at UCSA. A therapy for a type of congenital blindness called LCA10 that is delivering gene editing into the retina in the eye. And then also the first type of gene editing for a neuropathy that's caused by a disease called hereditary transthyretin amyloidosis, where a mutant protein that's actually produced in the liver causes neuropathy. And so their strategy is to try to disable that mutant protein in the liver, which should cure the neuropathy component, hopefully. So as you've heard, I think already from Dr. Scher and Dr. Wilson, many of the CMT type 2 diseases are the dominant type of diseases that I mentioned earlier, where the disease gene actively interferes with the function of the normal gene. And we've chose to focus on these types of diseases in which we also have strong evidence that one functioning copy of the gene is enough for a person to be healthy, in which case actually disabling the disease gene should be an effective treatment, which is the easiest way to use CRISPR technology in its current form. And so for this to be achievable, we have to achieve these following goals. We need to have the therapy be very specific for the disease gene and not the healthy gene. It has to permanently disable that disease gene. It has to either prevent or reverse the pathology that causes the disease in the neurons themselves. And then it should be generalizable to be able to treat as many patients as possible, and I'll talk more about what that means. And then finally, we have to be able to deliver it to the motor neurons in the spinal cord efficiently but transiently, so that it can do its job without causing long-term side effects. So I'm going to now show you the exciting results we have in addressing the first 4 of these challenges. So the first challenge is to make this treatment specific for the disease gene rather than the healthy gene. And so I'm showing you 3 examples where we've developed a potential CRISPR therapy for 3 types of type 2 CMT, type A, 2A, 2E, and 2F, 3 different genes. And all I want you to appreciate from this is that in each case, the target sequence that we're using to identify the mutant gene, which is shown with this red arrow, is only different from the healthy gene by 1 letter in the DNA sequence. But in fact, we actually can see that despite that minuscule difference, we are able to achieve here in these graphs where we want as much of this graph to be red as possible, showing that the genetic changes we're introducing are only affecting the disease gene and not the normal gene. And in fact, we can achieve 90 to 95% specificity so that we are, in the vast majority of cases, only hitting the disease gene. When we do these types of experiments, however, we're only looking at about 100 to 200 of the letters in the DNA sequence. But our total genetic code as humans is about 3 billion of those letters. So we also needed to be able to detect whether we might be making any changes in other places of the human genetic sequence that we weren't looking at. So my colleague Bika Weinert at the Innovative Genomics Institute developed a method that she calls Discover-Seq. And this was published 2 years ago in Science that can actually detect if there's CRISPR activity editing genes at any sequence without having to look specifically only in particular places. And we tested her method for our CMT2F CRISPR therapy for a particular reason, which is that we found that part of that gene, the HSPB1 gene, actually happens to have been copied into multiple different places in the human genetic sequence on different chromosomes. And so this is a high-risk situation that served as a good test case to see how her technology worked. And so we did a control experiment where we actually targeted the healthy copy of the gene, which is identical to another place on a different chromosome. And we found that, in fact, as we predicted, the CRISPR was causing changes equally in both the desired and in the undesired place in the genetic sequence, as well as in a third place that we didn't even predict. But when we used our disease-specific CRISPR, we could not actually detect any type of gene editing happening except for in the disease gene in the place where we wanted it to occur. Now I'm going to transition to what we've done most recently with CMT type 2E specifically, where we've used the human stem cells from a patient, actually from 2 patients with this disease. And we have treated them with the CRISPR therapy that I showed before for that gene. We've also used a specific genetic trick that we can do in stem cells where we've completely repaired the genetic sequence to a normal sequence, which I'm calling this corrected version of the cells. And then we've gone on and turned those stem cells that have been treated with the CRISPR therapy into neurons. And we are comparing them, the patient's own neurons from the patient's own cells with those from an unrelated healthy person and from the CRISPR-treated neurons, as well as these neurons that we have artificially corrected the patient's sequence to a normal one. And what you'll see on the pictures to the right is that the patients who have this mutation that causes CMT2E, their neurofilament proteins, which are highlighted or stained in green in these neurons, are building up in the middle of the cell rather than being transported down along the length of the projections or the axons. And we see that in these pictures, we don't see that abnormal accumulation either in our CRISPR-treated neurons or in the artificially corrected neurons. And so we've been able to show from this that the disease gene is permanently inactivated after the CRISPR treatment. We've done this in 2 different patient lines, one from a woman and one from a young boy. And to go on to make sure that we were not just cherry picking the best pictures, we've used computer software that automatically measures the neurofilament accumulation in this part of the cell and thousands of cells, and shown that in fact our CRISPR treatment here in white is equivalent, produces neurons that have equivalent outcomes to both the healthy neurons and the ones that are genetically corrected. So this is very exciting that we can make a CRISPR therapy that can essentially, at least in a dish from human stem cells, cure the disease. But the trick and or the next challenge, which I saw Dr. Scher mention this in his introduction earlier, is that not only are there many different CMT2 genes, but every CMT2 gene also has many mutations. So here I'm showing you the neurofilament gene, and this is not even a full list of all the different mutations that cause CMT2 type 2E in this gene. So going through this process and testing a treatment that's specific for every single one of these individual mutations is not practical. So we wanted to think about how could we do this in a way where we could make one treatment that could treat, if not all, at least the majority of patients. And so the strategy that we're using is actually to take advantage of the fact that we all have types of genetic variation that are essentially neutral. But that we could find in patients with CMT2, any form, in this case I'm using, again, CMT type 2E as the example, that if we knew what variants the patient might have that are neutral variants that we call SNPs, that just by chance happen to be linked to their disease mutation, we could potentially design a CRISPR treatment that would be targeting these sites of normal variant that could then treat any patient with any mutation in the gene that happens to have that variant. And it turns out that actually more than 40% of all people have a very common set of variants that are near the NEFL gene that could be used for this strategy. And the reason why this is useful is that we know, and others have shown, that you can use a pair of CRISPR enzymes and actually delete all of the genetic sequence in between. And so by targeting these common variants that are surrounding this CMT type, the CMT2E NEFL gene, we could actually delete the disease gene in any patient with any mutation who has those normal variants. And we call that DNA excision. So we developed a methodology to be able to very accurately measure how frequently DNA excision is occurring, and went on to show that that excision actually is more frequent and more efficient than people previously appreciated, especially when removing larger pieces of DNA. And we went on in our CMT type 2E patient iPS stem cells to show that we could actually make an excision of delete the start part of the mutant gene, and that even using a pair of these CRISPR enzymes, we could get more than 94% specific deletion of just the disease gene. And furthermore, this type of excision strategy is already being tested in that clinical trial for congenital blindness, LCA10, that I mentioned earlier. So we'll learn from their results. So in summary, I just wanted to, again, highlight what we're doing is taking patients with these three types of CMT2, making stem cells where we can design CRISPR gene editing that can make permanent genetic changes in those cells that converts the diseased neurons into healthy neurons. And the next challenge is how we can use animals to be able to design strategies to deliver that in the body so that these things can be tested in people. So I will end my show and stop sharing it now.
So thank you very much, Luke. That was great. I'll again hold the idea that we might have time for questions till the end of all of the three speakers. So we'll see how this plays out. And that brings us to Rob Burgess, who will speak to us from, I presume, Maine. Hello, Robert.
Hi, Steve. Yes, I'm in Maine. Thank you very much. Let me get my slides up here. This worked earlier. All right. Here we go. Can you see that all right? I can't see you, so I'm gonna hope that we're good. So thank you very much. As Steve mentioned, I'm at the Jackson Laboratory in Bar Harbor, Maine, and this is actually a picture of the Jackson Laboratory. The buildings in the foreground are the lab, and the background is Acadia National Park, so it's quite a nice place to do science. And I don't have a disclosure slide, but we're a nonprofit research institute, and I don't have any conflicts of interest. All of my work is funded by the National Institutes of Health, and as of recently, also by the Charcot-Marie-Tooth Association. So I want to give you a fairly high-level overview today of a few different projects in my lab and in a few other labs about more drug development strategies for treating type 2 neuropathies. And Dr. Wilson and Dr. Judge laid out some nice genetic strategies, but what I'm going to talk about today would be often more pharmacological approaches. So when I first started working on Charcot-Marie-Tooth disease, which was about 15 years ago now, a lot of the emphasis was on gene discovery. People were doing human genetics, trying to identify all the different genes that can cause all the different types of CMT. And now, through tremendous success in those efforts over the last couple of decades, we now know there are about 100 genes that can lead to different forms of Charcot-Marie-Tooth disease. So that sort of raises the question whether this is in fact 100 distinct diseases. And at some level, that's probably true because they're all caused by different genes and different mutations within each of those genes. But I think it's reasonable to expect that there are going to be some common mechanisms and some common biological pathways that are going to be shared between different types of CMT. And those common pathways might give common interventions, common drug approaches or therapeutic approaches that might be able to impact maybe not all forms of CMT, but at least multiple forms. In addition to those kind of I don't want to say generic, but broader approaches that might affect many different types of CMT. There are also more customized approaches or bespoke treatments for specific genetic forms. And some of those include the gene therapies and antisense oligonucleotides and CRISPR approaches that we just heard about, where you're really targeting specific DNA sequences or specific genes. But there are also, in some cases, drug targets that might be specific to a given form and a given gene and a given type of Charcot-Marie-Tooth disease. So in my talk today, I'm going to kind of work backwards here, and we'll start with some drug treatments that might apply to one or maybe a few types of Charcot-Marie-Tooth disease, and then talk about some therapeutic efforts that might apply more broadly, especially across different types of type 2 CMTs. So the first one that I want to mention is a recent discovery made by a whole team of people involved in CMT research. And this was the identification of SORD, sorbitol dehydrogenase, as a cause of recessive type 2 neuropathy. And this was just published a couple of years ago in Nature Genetics. And some of you might have heard Stefan Zuckner in an earlier session talking about some of the work on SORD. But I think the exciting thing about this is that there's a real possibility for treatment, because sorbitol dehydrogenase is a pretty well understood enzyme, and it catalyzes kind of a conversion of different types of sugar from glucose to sorbitol. That's mediated by aldose reductase, and then sorbitol to fructose is catalyzed by sorbitol dehydrogenase, or SORD, the gene that's mutated in these patients. So part of the problem in this disease is thought to be the buildup of sorbitol, because you've basically kinked to the pathway here at this final step of sorbitol to fructose. So if you kill this enzyme, you build up the intermediate sorbitol. But you could potentially prevent that buildup of sorbitol by inhibiting the first enzyme, aldose reductase. And fortunately, there are actually aldose reductase drugs, aldose reductase inhibitors out there. They're approved for use in humans in Asia and in a few other countries, but not in the United States. But there's now a real effort to get clinical trials going to determine if aldose reductase inhibitors are going to be effective in the treatment of SORD-associated type 2 neuropathy. So this is a nice one where the identification of the gene and what we know about that gene kind of led to an immediately good candidate drug therapy in aldose reductase inhibitors. In work from my own lab, we recently published a paper showing that tRNA synthetase-associated neuropathies activate a cell stress pathway that's called the integrated stress response. And it's through this sensor protein called GCN2. And there are several different tRNA synthetase-associated type 2 neuropathies. My lab jokes and calls these the pirate genes, because they're all called GARs and YARs and Rs and HARs. But we've been working a lot with mouse models of glycyl tRNA synthetase GARs, which model Charcot-Marie-Tooth 2D, and also tyrosine tRNA synthetase, or YARs, which models dominant intermediate type C. And in both of those cases, we found that the integrated stress response was activated very specifically in motor and sensory neurons, the cells that are affected by these diseases. And we found that if we blocked the integrated stress response, it was really beneficial. And I'll show you one piece of data on that. So this is from our recently published paper. And in all of these graphs, the black line are healthy control mice. And the blue line is our GARS mutant mouse, basically the model of Charcot-Marie-Tooth 2D. And then the red line is the CMT2D mice without GCN2. And in this case, we genetically deleted GCN2, but we also did these experiments using an experimental drug, and we got similar results. Not quite as good, but actually quite close. So here, just looking in the first panel, we're just looking at body weight, and you can see normally these mice have muscle atrophy. They're smaller. They're weaker than their littermate controls. But when we get rid of GCN2, actually the mice are pretty close to normal in size. This is a slightly different test of grip strength, similar to what Dr. Wilson described earlier, but we put the mice on a wire grid, basically chicken wire, and then we turn it upside down and we see how long they can hang on. And a wild type mouse can hang on all day. We stop the test after 1 minute, after 60 seconds. So the black line is the wild type mice hanging on for the full duration of the test. Our mutant mice are weak, and they usually fall off in just a few seconds. But again, when we get rid of the GCN2 gene, the mice now actually perform pretty well and actually get bigger and stronger over the course of this experiment, which was exciting to us. They also no longer have reduced nerve conduction velocity. They no longer have as much axon loss as we would normally see in the mice. The axons are still smaller. They're smaller than the healthy control axons, but they are larger than the axons when GCN2 is present and the integrated stress response is activated. And this is just a quantification of innervation of the neuromuscular junction, the synapse between the motor neurons in the spinal cord and the muscle in the periphery. And we have better innervation of neuromuscular junctions when we get rid of GCN2 also. So we think this is going to be an exciting possible therapeutic for the different tRNA synthetase-associated neuropathies. We're testing it in the Yars mice now to see if it has similar outcomes there. And again, GCN2, there isn't an approved drug now, but we're actively talking with pharmaceutical companies and biotech companies, and there are various efforts underway to try to develop a GCN2 inhibitor for clinical use. Another interesting potential target that's been mentioned a couple times today is SARM1. So SARM1 was identified about 10 years ago by Mark Freeman, and it was actually from work he was doing in Drosophila, in fruit flies. And what Mark found is that SARM1 is part of an active program of axon degeneration. So your cells are actually programmed to degenerate, which sounds a little counterintuitive. But where that can be helpful is in a situation like an injury. If you're in a car accident and you have a nerve crush, in order to facilitate the regeneration of those nerves, you actually need to clear out all the debris downstream of the injury site. And SARM1 is part of that active axon degeneration pathway. But it also sort of makes sense that since the major problem in Charcot-Marie-Tooth disease is axon degeneration, if we inhibited that pathway, that active program of axon degeneration, maybe that would be a good way to intervene to prevent axon degeneration in disease. And in fact, SARM1 inhibition or genetically deleting SARM1 has been shown to be effective for preventing diabetic neuropathy or preventing chemotherapy-induced neuropathy, for example. So we're testing that now in a variety of different Charcot-Marie-Tooth models. In my lab, we're using a SARM1 inhibitor to try several different 4 or 5 different mouse models of Charcot-Marie-Tooth disease. And there are other groups, Amet Hoke, for example, and Aaron D'Antonio and Jeff Milbrandt at Washington University who are doing similar work. And again, just to show one piece of data, and this is actually from Mark Freeman's paper. So this is a cross-section of a sciatic nerve in a mouse. And if you then cut the nerve, typically the axons degenerate downstream of that cut site. And that happens fairly quickly. Eventually, they'll regenerate reasonably efficiently, but within a few days of being cut, the axons usually degenerate. And here's a picture of the axons looking pretty bad after 2 weeks. If you get rid of SARM1 in those mice and then cut the nerve, the axons now actually look pretty good. They're preserved. They haven't degenerated. And that's true actually for weeks and weeks. So this picture is after 2 weeks, but it persists even longer. And these other pictures just confirm that even at the electron microscopy level, these axons actually look pretty good. And this picture on the right is neuromuscular junctions. So the synapse from the neurons onto the muscle. And again, normally the nerve, if cut, falls away from that synapse in just a couple of days, but it persists much longer when you get rid of SARM1. So again, SARM1 is an interesting interesting potential target for lots of different forms of axonal neuropathy where the primary problem is axon degeneration. And a lot of work going on right now to test just exactly how broadly applicable it may be in Charcot-Marie-Tooth disease. A similar kind of broadly applicable approach is Hdac6 inhibitors. And the logic here is that Hdac6, so this stands for histone deacetylase 6. So this is an enzyme that has a deacetylase activity. And the reason it's relevant to Charcot-Marie-Tooth disease is that tubulin in the peripheral axon is acetylated. And acetylated tubulin works better than deacetylated tubulin. And tubulin is essentially the railroad tracks of the axon that allow things to truck back and forth between the cell bodies and the spinal cord and out to the distal parts of the axon in your hands and feet. And if those microtubules are acetylated, they work better. And if they're deacetylated, they're less efficient in that transport. So we have a double negative here. If we inhibit the deacetylase, that's going to promote acetylation. So the logic here is that even if the primary problem in these mutations, in these forms of Charcot-Marie-Tooth disease, is not a problem with tubulin or tubulin acetylation, eventually, as the axon gets sick, axonal transport gets impaired. That probably leads to decreased tubulin acetylation. That probably leads to an even sicker axon. So there's kind of this feed-forward positive feedback mechanism going on. And if we could inhibit the deacetylation of the tubulin and hopefully promote axonal transport, hopefully that will just preserve the axons and axonal function longer. And that's in fact shown to be the case in several different mouse models of Charcot-Marie-Tooth disease, including 2F, a couple different groups that have identified this in CMT2D with GARS mutations, mitofusin mutations, CMT2A, and just recently also Charcot-Marie-Tooth 1A, a demyelinating neuropathy, also had improvement with Hdac6 inhibition. So I think the effect size here is not huge. This is not going to be a magic bullet that's going to prevent axons from degenerating or even promote their regeneration. But hopefully we can improve function and at least slow down the progression of some of that axon degeneration by inhibiting Hdacs. And then the final note that I want to make here is the possibility of improving synaptic transmissions, neurotransmitter release, and communication between the nerve and the muscle by improving function of the neuromuscular junction. And the suggestion is, to just remind you of the anatomy, we've got the motor neurons that sit here in the front half of the spinal cord and the sensory neurons that sit just outside the spinal cord. And both of those project axons out to the periphery, to your hands and feet and your whole body. And we usually think of Charcot-Marie-Tooth disease as being a problem with either the myelination or the integrity of these axons. But I think it's not unreasonable to imagine that if the axon isn't very healthy, its synapse and this connection between the motor nerve and the muscle is probably also not very healthy. And diseases of the neuromuscular junction or neuromuscular synapse are called myasthenias. Myasthenias lead to many of the things that you also see in Charcot-Marie-Tooth disease of weakness and fatigue and impaired motor function. So the idea that the neuromuscular junction is less functional in Charcot-Marie-Tooth disease is supported by work in mouse models, including work from my lab. And then recently, there's now a clinical trial going on, coordinated out of Ohio State University, to see just how broadly Charcot-Marie-Tooth patients actually have aspects of neuromuscular junction dysfunction or myasthenia as part of their disease. And the interesting and exciting part about that is there are actually drugs out there and also new drugs under development that are supposed to improve the function of synaptic transmission at the neuromuscular junction. And again, while this probably wouldn't promote regeneration or even necessarily prevent the degeneration of the axons, it should improve function, you know, of the axons that are still intact. So I just wanted to cover a few of these different areas today, and hopefully leave us a few minutes for some questions. But just to quickly summarize, then. I think there are going to be some disease-specific pathways that are going to be identified for all of these different mutations, and two examples would be the sorbitol dehydrogenase and using aldose reductase inhibitors, or using inhibitors of the integrated stress response in GCN2 to treat Charcot-Marie-Tooth 2D and some of the other axonal neuropathies that are caused by mutations in tRNA synthetase genes. Maybe some more general strategies like SARM1 inhibitors to prevent axon degeneration in general, or Hdac6 inhibitors to improve axonal function and axonal transport. And finally, I think this fairly new, but I think really interesting idea of trying to improve neuromuscular junctions, synaptic transmission, and the connection between the nerve and the muscle as a possible feature and also possible point of intervention for Charcot-Marie-Tooth disease. So that's it for my presentation, and I will stop sharing here if I can figure out how. And I'd be happy to answer any questions, and I think we have a little bit of time left.
So thank you, Rob. I'm going to ask Sarah to help us with the questions and to which speakers they might be addressed for the couple of minutes that we have here. So I see a question. Yeah, sorry, go ahead, Sarah.
We have a couple, and I'll start with showing the first one on stage. Do you think it is likely that HARS mutations will eventually be a part of the testing and potential drug therapies? I am a type 2O.
Yeah, that's very interesting. So we actually now have a mouse model of HARS, histidyl-tRNA synthetase also. We do actually see some of those same changes, the activation of the integrated stress response in those mice as well. So we're hoping that HARS is going to fit into the whole category of glycyl-tRNA synthetase, tyrosine, histidyl, alanine, and that they'll actually have a shared mechanism and would all be candidates for these GCN2 inhibitors or related kind of therapeutic approaches. So it's sort of early stages on that, but our preliminary indications with our HARS mouse is that there will be a shared mechanism there.
Awesome. All right. When do you think gene editing will be available to patients?
I think we'll send that to Luke.
Yes, that's a great question. I think, you know, it's a hard question to answer precisely. I would say that we're still a few years away, probably, from having these treatments being tested in patients. And the problem really is the problem of finding a way to deliver the gene editing to the spinal cord, but in a way where the CRISPR is only going to be active and present for a brief period of time to do its job. The viruses that Dr. Wilson uses are great at getting there, and for the gene therapy strategies, it works great, but they were really designed to have those things last in the long term, and we have to find ways to alter them or use other ways to get them there that's temporary, and we need to do what they're doing, which is to test them in like non-human primates, large animals, for safety testing first.
Fantastic. So it looks like we have come to time. So I'd like to thank all of our presenters. Wonderful type 2 gene therapy drug development discussion. We have one session left, which is our biotech panel. So please take a moment and join us in that session. It should be starting in about 5 minutes.
Thank you, everyone.